we are ready to go ahead and get started with our actual topic, which is Eyewire. And Amy, thank you so much for joining us. And it's such a quick turnaround from the previous event, so I'm glad you could join us. And here's the bio slide I showed at the very start, but as a reminder, and I'll let Amy introduce herself properly, but we're talking to the executive director of Eyewire and someone who is a crowdsourcing specialist, which is a cool title, by the way, at Princeton University. I feel like that's a very I don't think anyone in the world really has that title, so it's
Had to
unique,
make it
right?
up. They had to make it up for
You
me.
made it up? Oh, they
Yeah.
made it up for you. I guess that makes sense. Yeah, it's a new world. Why not? So, Amy, I'm going to go ahead and drop my screen share so we can actually see your face and say hello. But yeah, do you want to give yourself a little bit of a more introduction?
Yeah, sure. So hi, I'm glad to be here today. I love SciStarter, obviously. Who doesn't love SciStarter? Yeah, it's been a little bit of a crazy week, past couple of weeks for our research group. So I'm a part of Sung and Murthy Labs at Princeton University, specifically in Princeton Neuroscience Institute, where we are doing connectomics, which is making maps of brains at synapse level resolution. And so you can see all the connections among all the neurons, and this allows us unprecedented levels of modeling and understanding the role that like how actually neurons drive behavior. And so we involve citizen scientists in this through Eyewire, which is mapping a mouse retina and zebrafish hindbrain. In our most recent project, Flywire, which mapped the largest whole brain to date. It's a whole fly brain with 140,000 neurons and 50 million synapses. And it just came out October 2nd. Actually, I have it right here. I just got it. Especially with
Oh, nice.
nature. So we are very excited. There's 9 articles in this Nature all about the fly connectome, and then there's additional articles in Nature Communications and Nature Neuroscience, including the map and annotations of the brain, but also analysis of the connectome. And a fun fact: so the main articles in here have a lot of authors. Our main article, because we do big, collaborative science. Originally, it was with just citizen scientists, and now it's citizen scientists working alongside professional scientists. But one of the papers in here on the visual system that discovered 200 new types of neurons, the third author on that paper is a citizen scientist, is Christoph Crook. So if you ever watch this, woo, we cheer for you so loud in the lab. But yeah, citizen scientists
Awesome.
played a huge role in that. But yeah, so my background is really in citizen science and in crowdsourcing. Before working in citizen science, I was working in health and doing TEDx events, but that was a long time ago now. It's been over a decade doing citizen neuroscience, and we have exciting new projects in the works. So I'm just really happy to be here, especially at a time when we actually have a publication and some real important scientific results to share as a part of the contributions from the citizen science community.
Yeah, absolutely. Thank you for all of that information. That was awesome. And I do hope Christoph, when eventually, if he watches this, that he'll see the call out. And oh gosh, I lost my train of thought. What was it?
I'll send it to him.
Yes, please do. So this project, you started talking about a couple different projects that have come out of it. So I'm really excited to talk about those things because you are, or Eyewire and the whole system of projects is a great example of what can really happen with the science of citizen science, or rather citizen science becoming outcomes. Because we oftentimes, for everyone who is more familiar with other citizen science projects, oftentimes a lot of it is like long-term research. And even though that might be the case for Eyewire and Flywire and others, we've had really, really big impacts already. And that was starting, I think you told me it was Flywire's in 2019. So within 5 years, we have really big news to share. And so it's really exciting to see a project that's showing the beginning of high speed. You mentioned like collaborative, open work across an industry or a topic in science that just makes it a lot faster. So I'm excited to talk about those things. I'm going to have us back up a little bit to talk about where Eyewire came from to begin with. And so where the need came from and how it was developed. I'd love to hear more about that for the audience
Yeah.
here.
Sure. So, Eyewire, I think, is a great example use case of how AI can really transform things. It can transform the rate of progress and of discoveries. So, Eyewire was I don't know if it was the very first, but it was like the largest and a big pioneer in using AI to do what we call automatically segment out a data set. So we're working in connectomics where we want to see individual synaptic connections among neurons. And the synapses are so small that they're smaller than the wavelength of light. So you have to use an electron microscope to see them. So you take a volume of brain and you slice it very thin with a diamond knife called an ultramicrotome into these really thin layers. And then you take an image of each one of those layers with an electron microscope. And then when you stack all those images, you have essentially a volume of tissue, like a virtual volume of tissue in the form of like a stack of images. And the old way prior to Eyewire of reconstructing the neurons that have grown within this volume of tissue was people would go like slice by slice and color inside like a coloring book. And there's thousands, tens of thousands of slices to make up even a piece of one neuron. So it would take hundreds or thousands of hours to map one neuron. So that's obviously not scalable. Enter Sebastian Sung and, the world of computational neuroscience, which was pretty fringe at the time, but training AI to automate this process. And so to try to basically the neurons in the tissue, the edges of them are stained dark gray and the inside of the cell is light gray. So you're kind of training it to like start in a light gray area and look for the dark gray edge. And then you go, across an image and up and down through an image. And that creates this little 3D segment, which is why we call it segmentation. So now the human proofreading side that the AI still can't do automatically has been dramatically reduced. You don't have to go 40 nanometers at a time reconstructing neurons. You can have these 3D pieces that you put together to construct these whole neurons. So Eyewire kind of stemmed out of this. And in the early days of Eyewire, we estimated that it took like around 40 hours of expert human time to proofread one neuron, which prior to Eyewire, it could have taken 1,000 hours to map a neuron. So it was really an order of magnitude improvement thanks to AI there, but still 40 hours of expert human time is a lot. That's like a lot if you're trying to map thousands of neurons. So this is sort of the inspiration behind Eyewire. The output of this machine learning segmentation was these sort of odd shaped Lego blocks that you could fit together to make these oddly satisfying, like complete neuron structures. So this is what we kind of turned into a game in Eyewire and, made a whole training for it and, tried to communicate like why it is impactful, but also kind of how it helps research. That's sort of the origin of Eyewire. We had over the years a few 100,000 people from around the world sign up and map cubes of neurons across the brain. And in fact, actually, it's the most elite Eyewirers that were now contributing to the whole fly brain that was just, in nature. Did that answer your question?
Right. Yeah.
Sidetracked.
No. Yes.
Memory lane.
I think you did an excellent job of going back to the beginning. And so I have the images that you sent me earlier, and I'm actually going to share the one that has Eyewire on it, just for anyone who's having the thought of what am I supposed to be picturing here as a part of what the project looks like. And so I believe, and I've gone through some of the training, but I unfortunately have not gone through the whole project or like gotten to the point where I can actually earn points. And so when you're looking at the screen as like a participant in it, you're looking at these really goofy looking weird objects that are coming through as you're looking at the neuron or what's the right word? Neuron?
Neuron,
Part
yeah.
of the neuron, or is it the whole thing?
You're looking at, so in Eyewire, you have a little cube and it's like 4.5 microns on each side. It's a tiny volume of brain. And your challenge is to map a branch of a neuron from one side of the cube to the other. And that's the puzzle that you have to solve. And then we have you hit submit when you think you've done it right. And then Eyewire will assign the same cube to other people. And then it will compare the reconstruction. And if the majority of previously accurate players map the neuron to exit the cube at the same point, then the system will spawn a new cube directly adjacent to that first one. So we go cube by cube by cube, tracing out the branches of these neurons.
Oh, so it's almost like sequence probably isn't the right word, but it's almost like you're building the story outward. Like
It is sort of
you're
like
looking at the pieces.
Yeah,
Like a sequence. Okay.
yeah, and you got to follow, all the branch points of the neurons. You got to check all the ends to make sure there's not missing pieces. So yeah, it can, and as players level up, they can unlock additional data sets that have different, what we call segmentation. So that have, Eyewire launched first in 2012. So it was state of the art at the time, and it is still pretty advanced as far as the automated reconstruction. But the technology, of course, over the course of 10 years has had probably another order of magnitude improvement, which is why we're able to do projects like Flywire, where we're mapping an entire brain versus, a section of a mouse retina.
Right. So, speaking of it being sections, then does that mean that are we consistently adding new data to work on in Eyewire, or are we still proofreading the ones that have existed for a while? What is the
Yeah,
status?
that's a great question. So it's still being proofread. That's
Wow.
how
Yeah.
long it can take to proofread some of these data sets. Like the first connectome that was ever proofread was C. elegans, and it has 302 neurons, and I think it took 12 years to do, 12 or 15 years to map 300 neurons. And now Eyewire has mapped, I don't even know what the total number is, something like 5 or 6,000 neurons. And some of those are in the retina, and some of those are in zebrafish, which is a totally different species, different data set. Yeah, and then in the newer data sets, we're more than an order of magnitude higher with something like 140,000 neurons, which is kind of crazy.
Wow.
See how far it comes.
It
But
is crazy to think.
there's still neurons being mapped out in Eyewire. We kind of originally started in the middle of the retina, sort of like a long, very, very wide club sandwich. It has like multiple layers, and in each layer there's different, like kind of like in a club sandwich, you might have, I don't know what's in a like turkey and cheese on one side, on the other side like ham and lettuce and tomato, right? In the retina, you have ganglion cells and amacrine cells and bipolar cells, but they're found in very discrete places. So one of the ways that we've done it in Eyewire is we divide up the whole data set into these little sectors, and then we try to map all of the neurons from one layer that were found within that sector, and we kind of work our way around. But we are actually in the home stretch of a totally new retina data set. I guess I'm allowed to share this because I already have. Yeah, so we have a new we have a new mouse retina that has been acquired by Sung Lab at Princeton. So it's a whole mouse retina, I think, which would be a mammal's entire field of view. And this has really important implications for understanding mammalian visual systems, potentially for, treatments for blindness in humans, right? Because a mouse is a mammal that can see. You are a mammal that can see. A lot of overlap and similarity there. But yes, this new data set, I think, is supposed to be ready in the next 4 to 5 months. So we're planning to launch an Eyewire 2 with it. So they're big announcement.
Are there multiple mice being used for, like, I guess is it one brain that we're currently looking at right now, even if it's segments of it? It's just one mouse that was
It's
sliced
one
and diced.
Yeah, so in Eyewire, it's a retina, like, which is an extension of the brain in the back of the eye.
Hmm.
And it does a lot
Yeah.
of computation as far as like detecting objects and sensing movement and like which direction you or something is moving in. So a lot of things that you obviously don't have to think about, like which direction someone is walking in front of you, because a lot of those computations are done subconsciously and automatically, essentially, in the brain. And a lot of those are actually starting in the back of the eye before any of the signals even go down the optic lobe into the brain. So yes, in Eyewire, it's one data set from one mouse. So when we try to map, all the neurons from this section of the retina, but it's not the whole retina, and it doesn't even go all the way through it. So in this new data set, it will actually be the entire retina and all 3 layers of somas, going from photoreceptors to the amacrine layer, all the way to we call the ganglion cell layer, and the axons of these ganglion cells go out and form your optic nerve that communicates directly with cortex back here.
Gotcha. Now, some of those words are very new to me, but I think I get the general idea. Which reminds me to mention, if any of you have questions about specific things that are said, feel free to add in the questions. And Roland actually did that a second ago, asking about the actual players in the game. She's tuning in from Florida. Thanks, Caroline, who's on Facebook right now. She's asking, what's a common mistake that players make while playing Eyewire? So when someone's already in, they've done the training, what are the mistakes that happen, or the ways that you measure that?
Yeah, sure. Hi, Caroline. Hello. I hope you guys are all non-flooded down in Florida. Okay, so common mistake that players make would probably be well, there's really only a couple kinds of mistakes that you can make, but like what we call a missed extension. So sometimes, so the reason that the AI cannot automatically map out all of these neurons is because the dendrites and the axons do not have a consistent diameter. So in many ways, neurons are like trees, right? But in many ways, they're not like trees because they're not, they're growing in 3D space. And their branches, for example, would be very thin and very blobby and very thin again. I think even one of the images that I showed you, that I sent you, Emma's doing great, by the way. I sent her some images like 5 minutes before the call started. She works them in, but one of the images has a picture of an actual synapse, and you can see the branch goes from being very narrow to very wide to very narrow. And the reason that those synapses have a big bulge is because they're full of neurotransmitters, these little vesicles that send chemical information from one cell to the next. So all the
This,
little bulbs, that one, but actually I think there's one that's blue and yellow that may be a close up. You can probably even see,
oh,
yeah, that's right here. So this is a synapse between two neurons in the retina. And the narrow part of the branch the yellow one is an axon and the blue one is a dendrite. So neurons send signals out their axon, it goes in through their dendrite. And you can see where the branch goes from being very narrow to very wide. If you don't know that that's supposed to be there, it looks really wrong. So that's one of the most common actual errors that players make, is missing synapses or thinking that the branch ends, because these branches just get so incredibly narrow. And sometimes they get so narrow that as you're looking slice by slice, you can't even actually see where they go. And we would call like a jump point, where the segmentation may get so narrow that you can't even see it. So there's actually a little tiny jump in the neurons, but it's correct. When you look at 3D, you can see, okay, that's the same branch, it's going the same place. And that's a great example of where humans are much better at that. It's very difficult to define that task in that challenge to an AI. But a human can look at the 3D and see this, say, this looks right, this doesn't look right. So yeah, I hope that answered your question. Finding the boutons, the
yeah.
synapse, is the constant tricky point.
It does seem to me like this is one of the projects where you are thoroughly learning a lot as you learn how to play it. Like the amount of neuroscience that someone is gathering from being a player here is for anyone who's a science teacher in like high school, consider this as like a way to teach about neurons possibly. I'm not quite sure, but I feel like it would be a pretty useful tool. There's a couple projects that are used in that method, so it is interesting. Do you have like teachers in classrooms that work on this?
Oh yeah, there's a lot of classrooms that use Eyewire, and actually, for our new project Flywire, we just released Flywire Academy, actually just last week, which is a curriculum tool. It's a set of 3 worksheets and lessons for high school and for college educators. And in our new project, part of what we've done with citizen science is we were making a lot of like interactive visualizations of neurons for the game and for the proofreading and annotation, for the proofreading of neurons and the labeling of neurons, like identifying cells. It's helpful to have good visualization tools. So yeah, we actually created a whole curriculum that uses like interactive live views of neurons, that uses the connectome, so you can easily see what cells are like the up or downstream partners of any neuron in the whole brain. And you can ask really interesting questions about how signals go from A to B and how signals are computed with by neurons in the brain. So yeah, Eyewire is used in a lot of classrooms, and hopefully Flywire will be too.
Amazing. And we do have a couple other questions. Another one from Facebook from Darlene, the founder of SciStarter. Hi, Darlene. Um, she wants to know, and I have a follow-up question to this actually, because we talked to Pietro Michelucci recently with stall catchers, and he uses an algorithm from you all, or that you developed to improve stall catchers. So it might relate to this. Darlene's asking, given the success of this project, woohoo, and the acceleration of AI and machine learning, how close are we to mapping the entire human brain? Best guess.
Yeah, so that's a good question. The volume of just making the images to image the whole human brain at this resolution is like a zettabyte or something. It's something like equivalent to all of the internet traffic on entire planet Earth for a whole year. So it's a huge amount of data, which is still unsolved, which is one reason why actually there's a recently announced program from the NIH, is sort of an expansion from the US Brain Initiative. That NIH is the National Institutes of Health for anybody international who's watching. But this is called the Brain Connects program, and it's pushing forward a number of sort of large scale and circuit-based projects, but one of those is to have a whole connectome for a mouse. So mapping a whole mouse brain. And at this time, it's not feasible, but we're starting with 10 volumes, each of which is 1 cubic millimeter. And that may sound really tiny, but that's actually huge from electron microscope perspective. Each cubic millimeter brain can have over 100,000 synapses, 100,000 neurons, and millions, many millions of synapses. And different parts of brain perform different types of computation. So, and a mouse brain is about 1,000 times smaller than a human brain. So hopefully by doing these we don't really have any maps of like mammalian brain at this scale at all. There's one program, the IARPA Microns program, that we were a part of some years ago that took $100 million to generate 1 cubic millimeter of mouse brain. And that was the proof of concept that it could be done. That's how like new this whole field is. So now that we did one, now there's an order for 10. And then after that, hopefully within 10 years, we will have a connectome, or at least underway a connectome for an entire mouse brain, which would be the highest resolution of a mammalian brain that ever exists. Like we don't even know how many different types of neurons there are in a mouse brain, much less a human brain. But we do know that there's so much conserved genes and homologs and circuit behavior across organisms. Like even in a fly, I just learned a few weeks ago that like 60 to 70% of the genes in a fly are also in a human, or have a homolog in a human. Like, so that's so crazy.
Bizarre to think about.
Yeah, I know. Like, uh,
All very connected.
yeah. So I think that a lot of, Eyewire kind of helped establish this as a field of science, this idea of connectomics and being able, generating the technology that allows us to see the brain in this level of detail. And it is the level of detail that we need in order to understand the brain. You need to understand which neurons are connected to which other neurons, because that's how these cells communicate and compute. And it's really thanks to machine learning revolution, in some ways video games, right? GPUs that made much more powerful computers and enabled the machine learning revolution that has brought us to the point that we are today. But so hopefully within 10 years we'll have a mammalian brain model, which will, probably catapult neuroscience forward by like 50 years, just being able to see what's connected to where. And of course, every drug and therapy that makes it to a human has started in a mouse, essentially, or been tested in a mouse. So yeah, sorry, that was a long answer. I hope we have a human brain soon. There's also a human brain projectome project that I think is a part of the Brain Connects program. I should look a little bit more. Ned Talley is one of the like program directors for the Brain Connects program. And for the neuroscience minded folk, if you look up Brain Connects, there's a lot of really cool sci-fi sounding science endeavors that are in the works from the US government right now.
Ooh,
Yeah.
sounds scary and exciting. And there's a couple of questions about future help for illnesses of the eye, like glaucoma and strabismus. I'm not familiar with that one. But on that same line, before we get to that question, I am curious if, like, what was the reasoning for starting with the retina for the mouse? Or was there a direction of like we're starting there to move out, or what was the?
So that's a great question. And if we fast forward, if we go back to when Eyewire first launched, it was not clear that it would be a success. I had lots of people tell us that no one would ever willingly map neurons. But later we've got lots of, we have many papers. I don't even know how many came out of Eyewire. But because it's only one data set, we wanted to start from some part of the brain that did not have a huge amount of variety, of a lot of variability across individuals, right? So if you're looking at prefrontal cortex, especially a really small volume of prefrontal cortex, there's probably going to be some stereotypy, across individuals, but the actual computation that's happening there is really diverse, and it's getting all kinds of crazy inputs from all over the brain. Whereas the retina is getting input solely from what's coming through the pupil of your eye, right? It's getting photoreceptors, it's getting photons received by photoreceptors, and those are getting processed, 1, 2, 3, or 4 steps in the retina. So it's very easy to see what the stimulus is and what the neuronal response is to that stimulus. And so for a starting data set in a new field, where you don't really have a ton to compare it with, starting with a well-studied area and with an area that you can define where the stimuli is coming from very clearly, and that can, I guess, contribute to what kind of more varied brain structures you could study in the future. I think those were some of the main reasons that Eyewire started in the retina.
Makes a lot of sense. Yeah. I wasn't expecting that to be the answer, honestly, but that makes a lot of sense. Does that mean that when we start looking at like the papers that have been written and the results that we're getting from Eyewire, are we looking at trying to solve questions of common human illnesses like glaucoma and other illnesses of the eye, or has that been reached at all yet?
Yeah, no, that's a great question. I mean, no doubt the circuit diagrams of the mouse are reflected in the human retina. And it's actually been a long time since I've looked at human retinal research. So I don't know that I can answer exactly how Eyewire is making its way into clinical applications related to vision. I should do a little bit of a deep dive to see if it has worked its way, if it worked its way into treatments. But I mean, I have a cousin who has a congenital like blindness, where he's slowly going blind in his 40s, and so occasionally, he sends us these updates of like retinal implants where it's basically synthetic photoreceptors that can really somehow connect to the optic lobe and can work as basic like edge detectors, sensing like lightness and darkness, which is one way that you can determine whether or not you're going to walk into something, for example. So I wish I had a better answer for your question. I know that the science of clinical treatments for eye disorders is definitely moving forward, like all the fields of science, but I'll do a little researching, and if I find anything interesting, I'll send it to Emma and Darlene, and they can post a better answer than what I just gave.
Yeah, absolutely. We will. We'll send it around to our two friends who asked, Jim and where'd they go? Jim and thank you, Angie, asking those two questions. Amazing. Okay. So we've talked a lot about what Eyewire looks like. Oh, and speaking of actually, Caroline asked a question of these beautiful, colorful brain pictures. She's curious about the modeling of this, and honestly, I hadn't thought about that, but that's a great question. Like, how do we get these very fun? Like, this is beautiful.
Thank
Is this
you.
from Eyewire?
Thank you. I'm that one actually. That's from
Oh,
Pierre. It's from the IARPA microns cubic millimeter. It's the big test cortex data set. So these are actually the excitatory neurons of gray matter in the brain. They're in the brain of a mouse, but the human ones look almost identical. Like very hard to distinguish the human ones from the mouse ones. But yeah, a part of sort of generating these connectomic maps is you get these 3D reconstructions of neurons, and they're accurate to nanoscale resolution. So you can even go so far as to have like the surface topology of neurons. So I've always found the neurons very mesmerizing and beautiful and otherworldly. Plus, before working on Eyewire, I had only ever seen a textbook neuron. Like I hadn't seen what a real neuron in the brain looks like versus like the textbook neurons are like motor neurons down here. So we started doing 3D rendering of these. So we use Cinema 4D or Blender. Blender's free. And so you can just import like the 3D models of neurons and add a shader, which is like a 3D term for color, to them, and then drop a camera in there and you can make stills and animations. And actually on Flywire YouTube and Peer YouTube, which is PYR, there's a lot of animations of neurons, including an animation. This is just a frame from one of the animations, actually. So there's an animation that syncs up really nicely here. So if you like neurons, go check out Peer YouTube. That's where we've been stashing the animations away.
and you mentioned that was PYR, right?
Yeah, PYR. Yeah.
PYR YouTube. All right, just drop in the chat for anyone who was looking
Thanks.
for that. Um, Darlene from Facebook just made a comment about great explanation, jump branch, and how humans are better at spotting some things. Oh, right. Yeah. Looking at the AI related concept of we still need to teach the machine. We still need help doing those smaller things that computers aren't quite good at finding yet. When I was talking to Pietro from the Stall Catchers project, we were looking at an example of one of the stalls where for anyone who's unfamiliar, it's looking at brain mice blood vessels and looking at the flow of blood through the blood vessels to see if something is stalled, like clogged. And in some of their circled areas, they're supposed to be looking at the highlighted areas. That's the feature that I believe that's the feature that was data or information the AI and machine learning used in Eyewire to help with the stall catchers project. And it's still being improved, or at least from talking to the PHO, like we were able to like click on one and flag it as this is an odd one to circle. We're not sure if this is actually an area that should be circled, and it would keep improving. So the algorithm on Eyewire and being able to um, improve the algorithm that goes to stall catchers, is that what the participants are doing, or is that improving the algorithm from the formal researcher side? If that, I'm not even sure if I phrased that question right, but
No,
I'm
I
just
think I can. No, it's good. It's a difficult question to figure out how to ask. So
yeah.
I think I understand. And I mean, so maybe one of the ways that the actions of people proofreading neurons factors into the eventual connectome map. Not in Eyewire, but in Flywire, we went through several rounds of where you do like we call it ground truthing. So where you have an answer that is absolutely correct, and you distribute you pick some random sections from within the volume and you re-ground truth it, or so you do ground truth to get your first segmentation, and then you can factor in the corrections that were created by the human members, and then factor that back in and feed it out to a new segmentation that actually fixes a tremendous amount of the errors based on the actions that the people did. So it basically learns what kind of errors there were in the data set, and then you can run it back through and basically have an improved automated segmentation. The downside of that is that it does it for the whole data set, so it can undo some of your previous corrections. So there's always a catch-22.
In the favor of making it more accurate, or does it incorrectly mark things?
Well, it takes the whole, so it would resegment out all the neurons in the whole data set. So say you've proofread, 2,000 neurons out of your data set. It takes the proofreading that you did on those 2,000. You do this in set points of time, and we version it this way. So to create a new version of segmentation, you take all those 2,000 edits and feed it in, and then you get a new version of your 3D segmentation, but for the whole data set. So it will have issued a new reconstruction also for those 2,000 neurons that you proofread. And since the AI is not still perfect, it can actually then go back and like change your answer and make your answer wrong because it thinks it's correcting it,
Oh no.
but it's actually making your answer wrong.
Oh boy. And that is why we need as many people as possible going through these more, and like more eyes on these. The power of crowdsourcing, even though we end up getting into this cycle. Hopefully that's not like the
No,
most
we come up with workarounds, but yeah, this one is
good.
just one of the funny catch-22s of like you're doing an improved reconstruction process, but then it leaves you some fail in the history.
Leaves you with some issues.
Yeah.
So let's talk for a minute about the other projects coming out of Eyewire. I know we mentioned Flywire. I don't know how many people are super familiar with Flywire in the news coming out. There was an SNL brief sketch, if you saw it. It was like the weekend report part. Did you see that?
I did. I couldn't believe
You
it. What? This is
For
all
anyone
I
who didn't see it, you can look it up on YouTube. But it was the joke. I was very like surprised that that's the joke they made. But in any case, it was
could have done
really
better.
fun to be able to see that
Yeah.
on Saturday. None of you knew about that. It wasn't. You weren't reached out to about like accuracy. I mean, would they really care? I don't know.
Oh, like NBC reached out to us asking for some images, and then they told us, no, we didn't run a story. We were like, oh, okay, well, too bad. And then we saw it on Saturday Night Live, so we were like, well, that's kind of better than a news story.
That is definitely a good way to reach people. Apparently, it was great. So if anyone hasn't seen it, go and watch some Saturday Night Live soon. But you mentioned Peer. Do you want to talk a little bit about what Peer is coming from and how that interacts with those who are participating in Eyewire? Like, would they be going over to that project eventually, or is it isolated or like invite only? I know Flywire was you sourced awesome Eyewire people to go. So is
Yeah.
Peer going to? Is it the same method?
Yeah, so that's a good question. Yeah, it is somewhat under progress right now. So I can
Okay.
about kind of the two projects that came after Eyewire.
Yeah.
So Eyewire, actually before we launched a separate project, we launched a new project within Eyewire, to the elite, the top level players in Eyewire. So as you play the game, you start out just as a general player, and then you become a scout where you can go and search the branches for mistakes. And then you become a scythe, which is when you have so much expertise that you can overrule the community. So you can, sometimes the consensus is a mistake. And so the scythe can go into any section of a neuron and can correct the mistake, and they can overrule what everyone else did on that cube. And then the tier above that is mystic, and we called it mystic because we've sort of opened up a portal into a new brain, into a new species. And there was also a new AI with mystic. So in Eyewire, players do cube by cube by cube, to map the branches. And in mystic, which was from zebrafish, another model organism that's widely studied, you had this sort of little AI agent we called Misty, and you would claim a cell and then you turn it on, and then Misty would go and try to grow the branches, but you had to kind of watch her because like she would make a lot of mistakes and sometimes start growing the wrong cell. And if you could imagine it going from like one cell to the next, eventually like you could just start growing this horrendous like giant bush of cells and lose your initial cell. So this was sort of a new type of gameplay that was within Eyewire, unlocked basically by having a very high level of expertise and activity within Eyewire. And it was an important step because at the time, there was a lot of disagreement from the researchers of whether citizen scientists would be able to jump from one data set to another, from one species to another. Us as the citizen science project organizers were like, of course they can. They can do anything. And of course they could. They did. So they went in and mapped a bunch of neurons in zebrafish. Several papers came out of this. But that also kind of set the stage for a couple years later. I think in between this point, we had COVID came, which disrupted us a little bit. I had two babies, which also disrupted us a little bit. And then we had Flywire, where we were getting to the point where we were ready to invite citizen scientists. But Flywire is different than Eyewire because there is no double checking. There's so many neurons in the brain that we are not going to try to have everyone look at every neuron twice. It's just way too much, take way too long. So what we did was we basically put together a test where people had to go in and use this new tool called Neural Glancer, and it's different than Eyewire because you have a whole neuron. So you can go in, you have a whole neuron, and your challenge is to find all the branches, map everything in the neuron, make sure that the soma, the cell body, is attached to the neuron, and then you submit it as completed. But there also was like no accuracy check because people are doing a coverage of one. So that's actually why we only invited invited the top 100 Eyewires into Flywire. So, and I think when they were playing Eyewire, they weren't even necessarily aware that they were unlocking this invitation. But they ended up, the citizen scientists ended up proofreading over 18,000 neurons in the fly brain. There's only 140,000 neurons in the whole fly brain, so that's a pretty decent chunk. And then they also, for the first time, so in Eyewire it's just mapping neurons, but in Flywire there's an ability to add annotations or labels to cells because there's a lot of different types of cells in the fly brain and in a whole brain. Compared to the retina, there's not quite as many different cell types. Like in the fly brain, you're talking 10,000 plus cell types. It's a lot and a lot still to be discovered. Like one of the main papers in our publication was an annotation paper, and they discovered over 4,000 new types of neurons just like in the central brain of the fly, most of which we to this day still don't even know what they do. So really exciting time of discovery. But yeah, so the Eyewires came into Flywire and mapped all these neurons, and this feeds eventually into Peer, which is our new project. It was originally named Peer because we were going to do it on the IARPA microns, that first test case cubic millimeter brain, but that ended up taking so long to do. And because it was a first pass, it has a lot of imaging defects, which means that it's very difficult to proofread. Even for the best human proofreaders in the world, you can't always find the axons of the neurons. So in favor, we are actually, we generated some new data sets, which also took some time. But so Peer is going to launch probably within like 2 months, actually, with a new hippocampus data set of the mouse. Actually, we redid the landing page just for you guys, SciStarter. If you put peer.ai You can go and sign up to get on the wait list for when Pier does launch, and we promise not to spam you with anything. But so Pier is so named from pyramidal neurons, Pyramidal, and those are the main excitatory neurons of the brain. So yes, those in the background are a few uh,
Ooh,
are are a few
yeah.
pyramidal neurons, and they have different layers. So we're launching Pier with one hippocampus data set from a region. So a hippocampus is a part of the brain that's implicated in learning and memory and in pattern solving and pattern recognition. There's a famous neuroscience case, the case of HM, a patient that had part of their hippocampus removed and was not able to form new memories after that. So it was actually one of the origins of understanding what this part of the brain does. But we still don't really understand how it works. It's receiving a lot of input from different regions of the brain. And so Pierre's launching with a section of hippocampus called CA3. And then there's another section that will be coming hopefully later this year, ERC, which is the entorhinal cortex. And we are making an infographic to explain what this is in regular human being terms because I am
That's
very
good.
I was like, do we really have to say CA3, ERC? These sounds like I don't know, these sound like droid models. Like but
They're cool words though, once you do know what they are. I was gonna ask, this is a silly question, but I remember hearing that one, it's an old story of someone where we figured out a lot about the brain because they were in an accident related to dynamite and a like a bar being like thrown through their face. And so it removed a part of their brain. I don't remember if it's hippocampus, but it had to do with like their personality and their like not inhibitions, but they ended up being like a very they cursed a
rageful.
lot. I don't
Yeah,
remember.
I think
Yeah.
so I should know this case because I think it is a famous case, but I think that yeah, this person was unable to control their rage, right? They
Yes.
became like
Yeah.
very emotional and angry and trigger happy with their fury. I presumably
Yeah.
some damage to the amygdala or like nucleus accumbens or all these different regions
Maybe.
in the brain. I don't know. I should look into that case so that I
I
can answer.
just read it.
Yes,
Ah,
Gage.
Caroline on Facebook, Phineas Gage. Yeah, that sounds right to me. Recently listened to a video that or a audiobook that was talking about it where it was underneath the left cheek or something and out through the brain. So whatever is di I don't know. This man was able to get himself up and walk his way over to a cart to get medical assistance with a giant hole in his face and back of the head. So, and yes, Bob Hershon is confirming, very famous Phineas Gage. Yes. Do either of you remember what part of the brain it was? Caroline or Bob? I love how the SciStar team is like, we are going to be there to talk about brains. The most action I've gotten from them in a while. In these calls anyway. This is amazing. Um, yeah, but whatever part of the brain, it's fascinating to me because like we know these words from like grade school, but like when you the amount of the story that people will tell you where you're looking at a textbook and the period at the end of the sentence is someone's entire career, like that one sentence is like what they study for their like it's a way bigger story than what we expect. And when we think about the brain, we remember these terms, but there's so many others we don't know, and like the I guess connectome. I don't know what the best way to use that in a sentence is at this point, but it's a big there's a lot of stuff going on in the brain, so it's not shocking to me that we're not quite at the looking at the human brain yet scenario
Well,
that I'm not.
there is one connectomics data set with human brain, HO1, that was from an epileptic patient that was generated by Harvard. But it's an awesome data set, and you can see real human neurons, and they look just like these. But it's a very narrow stretch of brain, and it was from an epileptic patient. You know, sometimes in extreme cases of epilepsy, they'll remove the part of the brain where the seizures are originating, and so that is one way to have human brain tissue to image and to map out is from the byproduct of one of those surgeries. But I don't know, I think that probably by the next generation, I don't know, my kids right now are just turned 3 and just turned 5. And I really think by the time they're in school, their experience with learning about the human body and particularly the human brain will be just completely different than the way that we do it now. I mean, you know, when Eyewire first launched, it was hard to show like an interactive 3D neuron in a browser. And now we can visualize, you know, 10,000 cells at a time, right? You can use Google Street Map to see a 3D interactive view of any place on the whole planet. Like I think once these interactive visualization tools kind of work their way into classroom settings, I feel like it's gonna just not only unleash like a whole new generation of understanding about how the brain works, but I think with that will unlock innumerable questions that we haven't asked yet, or that kind of needed to be asked by people who are just learning it, as they're growing up. So I think probably the next 50 to 100 years in neuroscience are going to be like some of the most exciting times in the history of science, period, because I mean it's human brains that made science in the first place, right? I mean, everything in our world is a result of human neuron activity, right? Even if you're physically building something, like it's all coming from intention and muscle movement, and we're kind of like right on the cusp. We're starting to make these amazing discoveries about how the human brain works, and how neurons in the brain come together to create a mind and a thinking organism with perspective. And a fly is very basic, but they actually exhibit some degree of preference. Like they have the male flies sing different tunes like to their female fly. Like these are really basic, and we still don't even understand fully how all of those work. But with this rate of progress, I mean, I don't know, by 50 years we'll totally understand how human brain works, and hopefully be able to fix it in most of the ways that it goes wrong.
Oh, that is a great collection of thoughts to be closing out anyway, with our 5 minutes left, because that is the philosophical part of like what it looks like in the future is really intriguing to me. So I hope all of you are thinking about that and excited for the future. You're involved in some pretty cool things if you're looking into Eyewire right now. I'm going to share my screen one more time because we didn't actually look at all the images that you sent me. So I just wanted to show one just in case there was any thoughts you wanted to make sure were added about any of these before we close out.
Oh, yeah, sure.
And if you have any questions too, feel free to drop them in the chat before we run out of time. That would be awesome. Sorry, go
Yeah,
ahead.
these are the 50 largest neurons in the fly brain and the fly connectome. And neurons of the fly brain relative to mammal neurons are just absolutely huge and massive. Like the largest neuron in the fly brain has over 100,000 synapses. It's just really crazy. In a mammal brain, you typically have like a few thousand synapses on a single neuron. So even though they're dumber, they've got some pretty monstrous neurons. So yeah, that's
Yeesh,
all.
yeah, what does that equate to? Like, what does it mean to have more synapses there? Is that
It
understood?
Yeah, it receives a lot of input. It's actually these two cells are kind of sticking up at the top, receiving a lot of input from actually a lot of really teeny tiny cells, or a lot
Oh, wait,
and
is
so
it these chunks
yeah, those two at the top.
or
And there's also some in the optic lobes, which are on the side that have, I don't know, 50,000 synapses or something that panel the whole back of the eye, whole inside of the eye, and are major inhibitory neurons. They can literally stop a fly in its tracks. Yeah.
wow, okay. Okay, we saw that one. This is the last one.
Oh, I don't know. I just sent you guys a human brain because at the end of the day, we all want to know how the human brain works, right? Like flies are great. They're good model organisms. Mice, critical for all clinical applications. But from a philosophical point of view, and from Amy Sterling, like I love the brain. Like this is the brain that I love. And this is the brain that I want to understand how it works. And that I hope through citizen science, we can get a lot more people thinking about.
Applause for that. Love it. This is also a really pretty picture of the brain in general. It looks like it was made out of Play-Doh in a very artistic fashion. So I'm a fan. Our brains don't actually look like that, right? They're like gray and pinkish. Are they pinkish or just gray?
Sort of translucent, and so they would be
Mm,
like the color of your blood,
gross.
with like pinkish. Yeah, I know when I make the renders, I try to make them look too fleshy. I'm like, can I make it like look like it's out of a nice like Play-Doh or something? I don't know.
Yeah, it's pleasant. It looks like art, which I guess, I mean, the brain, that's where art comes from us. So I think it makes sense to represent it as art.
Yeah, and
Um,
that's the
all
cortex.
right. So what was it?
Oh, I was just gonna say, yeah, that's prefrontal cortex, like right behind your eyes. That is what sets humans apart from every other species on the planet. We have a much higher ratio of prefrontal cortex than the other
Mm.
species.
And that area is most known for.
Integrative thought, like problem solving. I mean, it's taking signals in from all
Gotcha.
over the brain and higher level thought. is all coming from prefrontal cortex. And we don't know very much about what's in there, to be honest, about how those neurons are organized. So goldmine for the future.
Yeah, we'll find out. We'll find out in the next generation, hopefully. All right, and I didn't mention this because we were just so on a roll with all those questions, and I'm grateful for everyone who added in their questions. But in order to do Eyewire, here are some instructions from the SciStarter end where you can go to the project page and get like a good lowdown on what all is involved in Eyewire, and then the directions are right there on that page too. So you'll go straight to the Eyewire website by hitting the visit button, and it'll ask you to register. And you'll just go into the instructive engaging tutorial, which is where I am right now, is in the tutorial. And then after completing the tutorial, you'll start playing, earning points, and helping us with the goal, the connectome. And you can get all these points. It's so gamified. All these projects that are coming out is so gamified, are so fun. All the gamers that are just loving it. We have a whole slew of them, and I'm so glad you all exist, because it's like, this is not just science and like volunteer work. This is, you're supposed to have fun, enjoy, learn, and have fun. And then I think that's, oh yes, if there were any leftover questions, I think we're at time, so I want to make sure. Oh, from our friends, Bob wanted to mention, oh, BTW, Phineas Gage's skull is at the Warren Anatomical Museum at Harvard. So you can visit the man who had a metal rod thrown through his face in an accident, who ended up with a lot of rage. So fun fact. Nice. Thanks, Bob. If you,
I need to go see it, I guess.
yeah. Oh gosh, yeah. You should give us your 2 cents on the whole thing afterwards. The thing I was going to say next. Oh, right. If you do have any questions, you can always reach out to info@scistarter.org, and we will make sure to get the questions out to Amy. On the website Eyewire page that I showed you the link for before, there's also an option to contact the project leader. So if you want to send your questions straight there, you can. And we'll make sure that those get answered. And Darlene wrote in Facebook for us, great conversation. I learned a lot. Thank you. So yes, that was the next thing anyway. Thank you so much for making the time to talk with us, Amy. We super appreciate it, especially since you've been like back-to-back things these last couple of weeks. So congratulations to your whole team. And we're really grateful to have the time to actually talk to you and see what's going on from the citizen science perspective too.
Thank you so much. My pleasure.
Sweet. And for anyone who needs to run away because we're at the hour, just an FYI, we have a bunch of events coming up with these Tuesdays, including a starting point for Citizen Science Month next week. So if you want to learn about how to get your community involved, or just get ready to plan an event or something, if you want to do more this April for Citizen Science Month, now's your chance. And you can just sit in and listen. And then we'll be talking about spiders right before Halloween. So if anyone cares about spiders or wants to learn about them, it's going to be all the fun about the Joro spider, which is invasive. And I'm not sure how we should feel about it, because it seems like we might not need to be too worried. And they're super fun looking. They're like bright yellow and scary. Um, and then the last one, oops. And then we're going to be talking about wildlife corridors. So if you're interested in that, and then we head into Thanksgiving time. So if you're interested in food, get ready for some sourdough science and others. So yeah, and all the items are on that blog page too for you. And that's it, I think. Yeah. So if you have any questions, you can always reach out to us. We'll make sure to get the questions to the right people to answer it. And we would love to hear from you. So Roland dropped in the chat a post-event survey link that you can use. It takes like less than 2 minutes to fill out, or less if you're super quick about it. And we'd love to hear your thoughts. But otherwise, we look forward to hopefully seeing you next week and the following week and the whatever week it is the next time for more events, because we love doing these. So thank you so much, Amy, for joining us. I appreciate you. And hopefully you get to calm down after this excitement of, I don't know, do you have a ton of more events?
No, I think this is the last event relating to the Connectome. Now on to the next data set.
Nice, sweet. Oh, speaking of, do you have the link for the Nature Journal, like the online version, or is it only through?
No, and it's all open access. Let me just hold on.
Okay. Yeah, I figured that might be a good thing to share right before. But for anyone who's heading out anyway, thank you all. I'll make sure it's in the follow-up email too. But I just realized that might be helpful. Oh, yay! Yes, sweet.
There we go. I just posted to everyone. So that's it. Nature built a whole web page for all the papers that came out in the Fly Connectome. So with a little summary of what each one does, and you can link to them, and they're all open access. So you should not need a Nature subscription to read them.
Awesome. Thank you so much. These are beautiful. more of the beautiful pictures and the models. Wow, amazing. All right. Thank you everyone for joining. I'm going to go ahead and stop our recording and Facebook. We'll see you next time.